Hello, it's now my pleasure to introduce Dr. Iraj Ali, CEO, and Dr. Ed Samuel, EVP Technical Operations of Achilles Therapeutics. This fireside chat will be around 25 minutes, and the audience can ask questions during the session by entering them into the questions box below. So to start off, thank you both for joining us today. Can you give us a brief overview of Achilles CNET platform and its manufacturing process?
Sure. Thanks for the invitation to talk and tell you a little bit more about Achilles. So maybe we'll just advance to the forward-looking statements, and I'd encourage people to review those in their own time. It's available on our presentation attached in our website. But maybe if we move into the overview of Achilles, particularly for those who may be new to the story, we are targeting clonal neoantigens with patented technology and think that we can do something new and novel in terms of linking mechanism and potency, which have been key features in defining the success of cell therapies to date. So maybe going through each of those pillars in just a little bit more detail. Clonal neoantigens are a novel class of antigen, a target present on all tumor cells and absent from healthy tissue. And we've, in the field of oncology, never had a target quite like that. So we think this can unlock efficacy. We have a unique capability in identifying that antigen. We are respectful that there are a lot of other people out there looking at neoantigens, but we believe that what we're doing is both unique and best in class compared to the field. And we'll talk a little bit more about that too. Together, we think that gives us unprecedented control over our drug and our product. That allows us to quantify, characterize, and track the tumor reactive component, both in the product before we dose it, and then once we dose it and track it in the patient. And that gives you that really important ability to look at the T cells, essentially killing the tumor, as you can see on the illustration on the left-hand side. And that is really executed with a world-class scientific platform. So where are we in terms of our activities? We have two open studies, one in non-small cell lung cancer, which we believe remains one of the most exciting and most challenging areas to do drug development in oncology, but has a very significant opportunity for patients and for shareholders, and in melanoma, the stalwart of IO. Those studies are live and recruiting, and we will be bringing additional clinical data out in the second half of 2023. So that will really come together with 15 to 20 additional new patients of data, looking at our CNET, that's the clonal neoantigen reactive T cells, across our lung and melanoma studies. We expect that we will have some patients that are in the melanoma study where our drug, the CNETs, have been used in combination with checkpoint inhibitor. We hope and expect to see additional confirmed responses. We have already seen a confirmed response in our studies, and that will allow us to really understand and unlock the mechanism by looking at the specific features of the tumor reactive cells that we are producing, and what makes a good product, and how do we make sure that we are delivering a process that can deliver the best possible chance of seeing a response in each of our patients. We continue, for that reason, to develop our bioinformatic platform, Peleus, which is, we think, the best in the world, and our process development to optimize that to ensure that we are getting the product features that deliver clinical response. With that, I'll hand over to my colleague, Edward Samuel.
Thanks, Iraj. Yeah, so just looking at the schematic of our manufacturing process here, you can see, patient on the left-hand side who comes in for a surgical procedure. This is where we collect our tumor sample that serves two purposes. We use that tumor sample to take very small samples for sequencing. That sequencing data is put through our in-house bioinformatics platform, Peleus. And that enables us to identify a unique set of targets to each and every patient. These are the clonal neoantigens. It's this list of clonal neoantigens that's unique to every patient that we have synthesized into peptides. So you see that kind of going on in the middle part of this cartoon. Whilst that is going on with our tumor sample, we're isolating tumor-infiltrating lymphocytes and storing these down for downstream use. We're also collecting 150 mils of whole blood from the patient on the same day as the tumor surgery, And we use this starting material to generate monocyte-derived dendritic cells. And that's the key kind of enabler to enable presentation of antigen through those peptides. So we bring those three elements together. That's the clonal neoantigen peptides, the starting TIL, and those dendritic cells in a co-culture, and that drives the selective expansion of our clonal neoantigen T cells, CNETs. So we then freeze that down, and that gives us the flexibility to ship to clinical sites as and when the patient needs to be dosed. In concert with that, we've obviously developed a kind of key technology platform around our potency assay, and this really serves two purposes and is differentiated in terms of kind of the classical TIL therapies, in that we're able to both identify and quantify the tumor reactivity in the clonal neoantigen T cell dose in every single patient product. So we know what's going into the patient, we know what's in the bag when they get dosed, but we're able to look at that in concert with the kind of immune monitoring, where we can track those CNETs in the patient through the ELISpot platform that we use. And that enables us to look at both engraftment and persistence. And you can see here a case study of one of our early melanoma patients here on the right-hand side with a dose of 16 million CNET. And you can see the expansion of our CNETs in vivo up to day 10, and then a contraction of those CNETs. And so that's really important to enable tracking. And I think also correlation with CNET dose, and not too dissimilar what we've seen with the kind of very early CAR T studies. And so, we want to be able to test, bigger CNET doses and drive the kind of area under the curve here to get longer term persistence. And so both product release on CNET dose and in vivo tracking of our products is a kind of key differentiator for our potency assay platform at Achilles. In terms of our manufacturing facilities, we have two facilities currently manufacturing for our first in human studies at the Royal Free Hospital in North London and the Cell and Gene Therapy Catapult in Stevenage. And these serve our global clinical studies in the US, UK, and Europe. We're also identified a partner in the US on the East Coast in Philadelphia that will kind of enable us to kind of build out our capability in the US. And of course, in time to look at bringing all of that together under one roof in our own manufacturing facility. So I'll stop there, and perhaps we can move on to the Q&A section.
Great. So for your manufacturing platform, you've implemented a number of changes in that process. Can you walk us through the changes that you've made and the rationale and how they've impacted the product?
Yeah, absolutely. So, as we've kind of transitioned into process 2, which was very much built around kind of boosting the number of CNETs in our manufacturing process through a polyclonal stimulation step, we've now really focused very hard on the kind of TIL extraction step. So being able to get more TIL out of our patient samples that we know are very variable from patient to patient. So very much focusing on things like optimized culture conditions, being able to look at things like cell counts as we have those cells in culture that determine when to harvest our TIL from the patient sample. So I think that in kind of combination also with things like automation and introducing automation harvesting devices really enables us to make sure we're getting the most TIL that we possibly can from every patient sample that flows downstream to our kind of co-culture process and gives us the best chance of making the CNET doses that we want to give to our patients.
Got it. So beyond that, are there additional levers that you have at your disposal that could further increase cell number or modify the phenotype if needed?
Yeah, I think, as we begin to look at the kind of evolution of the process beyond that TIL extraction step, I think, if you look at the co-culture part of the process, the opportunity again to kind of further optimize things like our, kind of dendritic cell manufacturing, also looking at, the kind of presentation of antigen by the DCs, are key areas of interest, and also looking at other opportunities with automation in the kind of cell culture and bioreactor part of that manufacturing process. So, although we've kind of tied down a number of areas to ensure we standardize the process and make it more robust, I think there's, further opportunity to kind of refine the manufacturing process.
Got it. That makes sense. So for autologous cell therapies, vein to vein time, or in your case, tumor to vein time is an important consideration. So for your process, what is the time frame that you're currently working with, and What can you share regarding how that's broken down between the different steps?
Yeah, so for the manufacturing process, targeting, by the time we get to kind of commercial delivery of, 8 to 9 weeks for putting from procurement to having a product in the freezer and released ready to go. I think there are a number of areas that we're again looking at to kind of get to that 8 to 9 week target. Things like peptide manufacture. So at the moment, we very much reliant on kind of third parties for making those clonal neantigen peptides. And that's an area that we can exploit with the right investment in time to kind of bring down the turnaround time for making those peptides. I think there's also a number of other opportunities, and we've mentioned kind of automation, automating the cell culture part of the process, shortening cell culture times, and alongside that, kind of digitization is probably the third key area that we think again will enable us to kind of move with greater speed through the kind of the QA and QC and the release part of the manufacturing process.
Got it. That makes sense. So can you talk a little bit about the kind of analytics that you use to understand the process? I know you have some slides regarding the potency assay and things like that. Expand a little bit on what you see as the important parameters from your perspective.
Yeah, absolutely. So yeah, we've kind of focused in that slide just to kind of give you some visibility of the potency assay platform we have that is the kind of central pillar to being able to understand both the active drug component and then in the patient, and that's done with an ELISpot assay that we currently use with our translational science immune monitoring team. I think you then couple that together with a number of other technology platforms, flow cytometry to enable a very extended look at the product features. So we're interested in looking at markers for T cell fitness and memory phenotype and function, and those things together are really important to get a kind of greater understanding outside of the kind of key kind of GMP QC release assays. So building a greater understanding of all the T cells in the bag that gets dosed to a patient. I think the next level of precision comes we're looking at things like TCR sequencing at a single cell level that enables us to kind of really dig very deep into the analysis of those T cell clones in the product and then tracking in the patient. And all of those things together, being able to look at that in concert with kind of the clinical data to understand, what are the product features that really that we can look at to determine kind of clinical responses. So I think, broadly speaking, those are the kind of three key areas. But in addition, probably worth noting that we use kind of, circa up to kind of 200 clonal neantigen peptides in our manufacturing process. And the ability to be able to, again, deconvolute those, 150, 200 peptides to understand which ones are driving the expansion of the T cells in our co-culture system. And so, again, we have a number of assays, a matrix of assays that enable us to identify single peptide reactivities. And that's really, really key when you're looking to then bring all that data back in with the clinical data to understand how do you refine things like our bioinformatics platform, Peleus, and also, kind of hone in on those product features that we really want to ensure that, we have in every single product.
That makes sense. So from your perspective, what are regulators looking for with regard to product characterization on this front?
Yeah, I guess we've seen the kind of regulatory field move quite fast, or having to move quite fast as the industry kind of has more products, coming into clinical studies and onto the market. And I think, one of the key areas for us really is the kind of mechanism of action of our product. And I think the ability to be able to determine our active drug component, look at that in the patient once they've been dosed. And, again, with the things I've mentioned previously around the product features, really understanding what is it that's driving, kind of tumor remediation and clinical responses. So, we have the breadth of technology platforms to be able to determine that. I think you're kind of looking again with, how reproducible and robust the manufacturing process is. So, standardization and understanding your starting material. So I think, as you kind of move through the clinical development pathway, understanding what is it within your starting material that again would give you a very early indicator of kind of a successful product, a product that contains all the features that are kind of synonymous with patient responses. So I think building out that matrix of data surrounding, product characterization, product features, but then also, and that's a key differentiator for Achilles, is understanding, what is the active drug component. So I think there are probably kind of two key areas that we're focused on, and I think that's against the backdrop of a lot of interrogation around potency assays that we've seen in the field.
Right. And so in terms of your manufacturing capacity, what is your current capacity in terms of, say, doses per year or per month, and what kind of capacity do you envision you might need for commercial supply?
Yeah, great question. I guess the commercial supply, you're kind of certainly talking about delivering kind of thousands of products annually, both in Europe, UK, and in the US. Where do we stand today with our manufacturing facilities, the two facilities that support our clinical studies? Kind of circa 100 doses dose capacity, currently today, and I think at peak capacity would be able to move that up to about 150 or so. I think developing your manufacturing footprint and building out capability, like you described, with the US kind of CDMOs and internal manufacturing facilities, is something that comes with investment, but you're going to have to be able to reach the kind of patients with those kinds of tens of thousands of doses at some stage in the future. And I think, beginning to understand how the manufacturing process can evolve and become more streamlined will be an important part of that kind of delivery.
So on that note, you did mention automation a number of times. Can you elaborate a little bit on how you can incorporate automation or things like AI, everyone's favorite buzzword these days, in ways that can help address some of the manufacturing challenges?
Yeah, absolutely. I'll leave the AI question to Iraj, but happy to pick up the kind of focus on the manufacturing part. So I think one of the key areas that we identified with, that required that automation step is how we process our tumor samples. So, currently very kind of manual operator dependent. So you're collecting a tumor sample and setting that up into small pieces for culture. And that's something that happens in an open system. And the drive at Achilles really is to have a fully closed manufacturing process from the point of procurement at surgery to the point you put a drug product in a bag. And so we're looking at, introducing automation for that upfront tumor collection and processing. And that's something that we hope to begin to look at kind of in our kind of preclinical studies later this year. That will be a significant, kind of step change, I think, for the industry to close that type of element down in our process. Talked very much about one of the kind of here and now pieces, which is introducing, kind of automated harvesting devices, again, removing the need for kind of lots of those manual operator steps that are often associated with some variability. And I think, again, in the future, how do we get better at kind of the cell culture aspect, introducing technology, whether it would be sensors to look at things like glucose and metabolites, to understand specifically for autologous cell therapies, what's happening inside those cultures of their patients' T cells, and being able to react accordingly to harvest early or let the cultures kind of take a bit longer to expand. So I think those are broadly kind of three key areas of automation that we're very much focused on.
Yeah, so maybe I'll briefly pick up the point around AI and how that's used in our manufacturing process, which is in the target selection. So we are firm believers that the quality of the target is what differentiates Achilles from other neoantigen companies. And what we can do with confidence now is do two things. One is find targets that we believe are truly clonal, and that we can find targets that are highly immunogenic. And those are the critical features that you'll need to see clinical responses. And we use AI in both of them. I mean, AI in its simplest sense is allowing you to rapidly process very large data sets and allows you to essentially train your algorithm against a ground truth. So it tells the machine, we want to see this out. We like the clonals that generate an immunogenic response. Is there a pattern in the peptide? Is there a pattern in the way that peptide might bind or dock with an MHC that can be used to predict that a peptide is a better, or an antigen is a better peptide than another one? And that takes very, very large data sets, and invariably machine learning and AI to train your algorithm to get better and better at it. And what does it do in its simplest sense? In the simplest sense, we think that in a relatively small number of antigens, let's say around 30, so this would be a typical number that you would see in a vaccine, and certainly a sort of number we'd like to use in a cell therapy, a much lower number, we can accurately predict about 70% of all the antigens that will generate a T cell response. So this allows us to compress the process in terms of material, cost, and increase the robustness and delivery of the process. And we think, that's very much going to be the future. Can you target the best antigens with the sort of fastest and cheapest process?
Yes, that's great. So on the topic of cost, actually, in the current financing climate, cost is something that's top of mind for many. Can you talk about how the current environment that we find ourselves in has impacted your manufacturing priorities? Yeah,
currently the company is well capitalized. We have $173 million at the close of the last quarter, and we're in a fortunate position to have a very strong balance sheet that allows us to deliver our studies as we've planned. And really, we take a cautious approach to how we think about expanding the manufacturing footprint, subject to the company's needs, which will be driven by a positive data signal. So Ed touched on our current footprint that allows us to generate sufficient capacity to deliver our first in human studies, which is not insignificant. And as we sort of try to reach the numbers that we would need for a commercial launch, which have thousands at the end, then you would look to expand your manufacturing capacity and indeed attract either partnership or further capital to do that. So I think we're in a world where use of proceeds is extremely important, and all companies like Achilles are going to be extremely prudent and careful with the use of those resources. But our cash runway right now is through to the middle of 2025, and we feel that is a strong position to deliver our studies and positive clinical data.
Mm-hmm. Great. So in the last couple of minutes, I did want to ask what you think people might underappreciate about Achilles manufacturing tech and capabilities as you guys are in 2023.
Yeah, it's a great question. I think maybe the whole sector is currently underappreciated, but I think specific to Achilles, I think the principle is that all neoantigens are not created equal. I think the targets have very different levels of quality, and Achilles, I think, has the unique position in identifying both true clonals, and we can demonstrate that with the largest data set anywhere in the world, the TRACERx study, and immunogenic antigens that we have very high confidence will find the corresponding T cell response. And that's key. In the end, the targets are really only important if you can find a T cell. And we think our position in the field is unique through our exclusive access to the TRACERx study and through the exclusive IP and proprietary know-how that we've developed over the last 5 to 6 years. So I think that's number one for us. We're really a company about identifying quality targets. Number two, I think we have one of the most sort of robust and sophisticated processes in the TIL field. And we've shown that in our ability to characterize the key features of our product, both pre-treatment and post-treatment. And I think together, that's what's going to unlock efficacy for patients and value for shareholders, which is the ability to go after the best targets. We can do that currently with a TIL-based process. We can also do that with a vaccine, or even go after shared antigens with a number of allogeneic approaches. So we think target is king, and we have a unique position there. And in the area that we've chosen to pursue, at least initially, which is cell therapy, we think we've shown that our process is at the highest level of sophistication in the field of TIL-based drugs.
Fantastic. So I see we're at the end of our session time, so I'd like to thank you both again for the participation and the great discussion.
It's a pleasure. Thank you very much.